Interfusal muscle fibers are the skeletal muscle fibers that serve as specialized sensory organs, precipitators that detect the amount and rate of change in length of muscle. They are innervated by both sensory and motor fibers. Extrafusal muscle fibers are the skeletal standard muscle fibers that are innervated by alpha motor neurons and generate tension by contracting. Therefore, they will allow skeletal movement, which generates everyday movement and some exercise. For example, you need this in gymnastics, which is a sport which allows them to perform in their different movements. Gamma motor neurons control muscle contraction in response to external forces acting on the muscle. In response to these external forces, the gamma motor neurons induce the involuntary reflex movement called the stretch arc. The stretch arc is another factor within the muscle spindle apparatus region, which can be a synaptic reflex, which produces an automatic regulation of skeletal muscle length, whereby the signal entering the spinal cord arises from a change in muscle length or velocity. When a muscle lengthens, the muscle spindle is stretched and the nerve activity is increased. The stretch shortening cycle, which is SSC, refers to the muscle action which happens when the action muscle lengthening is immediately followed after by active muscle shortening. This combination of eccentric and concentric contractions is one of the most common types of muscle action during locomotion. The stretch shortening cycle is comprised of three phases: eccentric, amortization, and concentric. General elastic energy is the mechanical potential energy stored in the making up of a material or physical system, as it is subject to elastic deformation by work performed upon it. Elastic energy occurs when objects are impermanently compressed, stretched, or generally deformed in any matter. Plyometrics are exercises in which the muscle exerts maximum force in short intervals of time, with the goal of increasing power in the muscles and speed to strength. The function of the Golgi tendon organ is basically the opposite to the muscle spindle, which serves to produce muscle contraction. A proprioceptive sensory receptor organ that senses change in muscle tension. When a Golgi tendon organ is stimulated, it causes its specific muscle to relax by interrupting its contraction. When a muscle is inhibited by a Golgi tendon organ, the process is called autogenic inhibition. The function of the Golgi tendon organ generally can be thought of as opposite to the muscle spindle, which serves as a produce muscle contraction. The stretch reflex is a muscle contraction which helps in response to stretching within the muscle. It is a monosynaptic reflex which produces automatic regulation of skeletal muscle length. When a muscle lengthens, the muscle spindle is stretched and the nerve activity will increase. Autogenic inhibition refers to a reduction in excitability of a contracting or stretched muscle that in the past has been solely attributed to increasing inhibitory input arising from the Golgi tendon organs within the same muscle. Now on to energy systems. So this is the role of ATP in muscle contraction and resynthesis. ATP is a chemical energy store which produces energy. It provides energy to drive many processes in living cells and is mainly produced by mitochondria, which uses oxygen from aerobic respiration to produce this store. The scientific name for ATP is adenosine triphosphate, and it is the biological way to store and use energy and allow the muscles to be able to function, because it wouldn't be possible without an energy source. In the cell, it needs to get energy. The bonds between ATP break to form ADP and a free phosphate molecule. There is a theory called the sliding filament theory, which explains how energy is produced from ATP. The breakdown of ATP releases energy, which then allows the myosin, which is in the filaments of muscles, to pull the actin filaments inwards, and so shortening the muscle. This process happens across every myofibril in the muscle cell. The myosin then is caused to detach from the actin, and the cross bridge is broken when the ATP molecule binds to the myosin head, and this is the sliding filament theory, which involves ATP. ATP is mainly held together by high energy bonds. For this reason, making these bonds requires a lot of energy for them to be made, and therefore the process is endothermic, and it takes in energy to produce the bonds. The opposite is when the bonds are broken, which breaks the bonds and therefore releases energy, which is an exothermic reaction. After the energy is released due to the breakage of the bonds in the ATP, there is a need for the ATP to be resynthesized after the breaking of it so that it can work again. As energy is released when ATP is broken down, energy is therefore required to resynthesize it. The byproducts of the breakdown are adenosine diphosphate, which is also known as ADP, and PI. Cardiac muscle response for contraction of the heart allows the blood to pump through each of the four chambers, right and left atrium and right and left ventricle, and therefore supply the body with the oxygenated blood it needs. A pacemaker is found in the upper right atrium, which sends out an electrical impulse into the vessels, which there triggers the contraction and relaxation in order to maintain the movement of blood. In a marathon run, this action is incredibly important, as the heart will need to keep contracting quickly in order to pump oxygenated blood around the body to the muscles, because the mitochondria can then produce ATP energy, which enables the person to keep running. ATP is very important because it transports the energy necessary for all cellular metabolic activities. Phosphocreatine, along with the lactic acid system and the aerobic system, works by producing energy. Like a rechargeable battery, it allows the muscle contraction. PC stands for phosphocreatine, and it is a molecule that allows quick mobilizable reserve of high energy phosphate, which simulates energy in the skeletal muscle and the brain to recycle adenosine triphosphate, which is ATP, which is the energy currency for the cell. The muscles of the body use phosphocreatine in the first few seconds of an intense muscle contraction. This can be in shot put, for example, where a sharp burst of energy is needed to propel the put with the maximum amount of energy. And another example is sprinting. A lot of energy is required for just a short amount of time. However, the body is required to excel quickly, which needs a large and quick burst of energy. Unlike aerobic contractions, which use oxygen to produce energy itself, phosphocreatine uses the production of energy without the input of oxygen, therefore it is an anaerobic process. Phosphocreatine splits due to creatine kinase in order to produce ATP energy and therefore allow the faster actions to occur, as the braking lets out ATP, which is the energy, and therefore causes muscle contraction. There is enough PC in the muscle cell to continue to resynthesize ATP for approximately 8 to 10 seconds of physical work. The advantages of this system is that energy is released quickly and no waste products are formed. There are many different demands in the test which were performed. The exercise causes the heart to pump blood into the circulation more efficiently, as the result of more forceful and efficient myocardial contractions conducted due to the need for more oxygen delivery to the muscles, increased perfusion of tissue and organs with blood, and increased oxygen delivery. Aerobic exercise trains the heart to become more efficient. The lactic acid system has the ability to release energy in order to resynthesize ATP with energy without any input of oxygen, and therefore this process is called the anaerobic glycolysis. Glycolysis means the breakdown of carbohydrates causing the formation and build-up of pyruvic acid and hydrogen ions. Anaerobic glycolysis is the transformation of glucose into lactate when small limited amounts of oxygen are available. When sufficient oxygen is not present in the muscle cells, for more oxidation of pyruvate and NADH produced in glycolysis, it's regenerated by reduction of pyruvate to lactate, and therefore it can work again. When the ATP and PC stores have run out of energy, the needs of the body that are acquiring and provided by the lactate system. This system is reliant on the breakdown of glucose, which have been stored in the muscles as glycogen. When this is broken down, energy is released in the process, and this process, when glycogen is broken down, releases energy called glycolysis.